It's bigger than biotech.
Hello, and welcome back to another episode of It's Bigger Than Biotech. We have a very, very special guest with us today, Ron Demuth. He's a very intriguing person. I've already had one conversation with him. It has been genuinely the most entertaining and the most informative conversation at the same time. So thank you for being with us, Ron. I really appreciate you taking some time out of your day to speak with us on the podcast.
My pleasure.
Perfect. So, just to get us started, Ron, could you tell us a little bit about your role at Fluxion Therapeutics, Deal Ventures, and your other commitments?
Yeah. Let me begin chronologically. So, a very long time ago, I grew up on a grain and dairy farm in Northeast Iowa. But I was convinced at an early age that I was interested in science, and I certainly was not interested in farming. But as I progressed through my undergraduate career, I became increasingly intrigued with the neurosciences. And fortunately was able to, in my junior and senior year, work actively in a neurobiology lab. Jim West, who was an assistant professor there at the time in his electrophysiology lab, but we were actually doing work on prenatal alcohol exposure. And so it was a very cool study. We did some electrophysiology dosing in rodents, but also looking at the changes in anatomy of fetuses exposed to alcohol in utero and how their brains developed. Long story short, I was very fortunate with Jim West and a director level scientist at the NIH. We were able to publish that work in Science Magazine, which was great for me in terms of my future career. But it was intriguing in that it was the first concrete evidence that prenatal alcohol exposure changes the development of the brain. Without going into the details of it, it was a surprising effect compared to what we had anticipated, but that completely hooked me on science. If I had any doubt from that prior to that event about whether I wanted to go into pure research versus a practical applied background, because I was the child of farmers, so everything was only important in my upbringing if there was an application for it. But I was convinced that pure research is what I wanted to do. So I guess the question is then, how did my career evolve from being raised in a basic science discipline and planning a career for pure research to moving into operating, managing, and ultimately creating and building companies in biotech, biopharma, health tech. And I'm still doing that today. The question you asked today was really, so what was the evolution of going from that into science as a business, which is what I do now? And in particular, how did I get into the realm of investing in and growing early stage drug development and health tech companies, which is where I got to. One of the things I did in my early career was I was a research scientist at the University of Chicago doing pure research after graduate school, initially working for Rainier Guillory. Ray Guillory was a developmental neurobiologist, very famous for his work in the development of the visual system. And I mention Ray because he was my first, maybe my second true mentor in science, probably my second. I think Jim West at the University of Iowa, where we did that work on prenatal alcohol exposure, was my first real mentor. But Ray was an extremely accomplished senior scientist, full professor, director of the Center on Neurobiology, and taught me really how to ask critical questions. So I spent in total about 17 years at the University of Chicago doing research and teaching and then going into academic administration. But one of the things I was involved with there at the University of Chicago was building a whole genome sequencing center together with a group of senior postdoctoral students. While we were building that, everyone else was excited about sequencing the human genome. but we decided that we could do whole genome sequencing for fun and profit. And we launched integrated genomics in Chicago. We were one of the earliest biotechs focusing on whole genome sequencing. We did not sequence the human genome. We sequenced the rest of the economically relevant biosphere, and that was producer strains for fermentations, and that went across everything from traditional yeast fermentations, animal cell fermentations, bacterial fermentations, and even insect cell fermentations, which, as you may know, is the basis for a major part of the nutritional and vitamin industry. Those are largely produced, vitamins and all the rest of that. Many antibiotics are produced by fermentations. So we had a great set of customers in the US, agribusinesses, agro-processors, and in Europe. And at the same time, the Department of Energy and the Department of Defense were very interested in sequencing potentially pathological strains. So we did a lot of that work. Had a great time, made a lot of money. We're preparing to launch it for IPO near the end of 1999 to 2000. And then, as people that have been in this space as long as I have know, we had a tech and biotech meltdown and nuclear winter beginning in 2000 through 2002. Wow. And so the whole economy in that space collapsed. And near the early part of 2002, we lost the company. I was general manager there, and I was the one that invited people to stay home, stop showing up for work, and turned out the lights when we had to close the company. So I thought I was done in science as a business because my first foray was a failure. In a sense, it was. We built some great technology, some great software, but survived for about 6 months doing consulting to other biotechs, which were encountering similar problems. But I got a call to come out to the West Coast of the United States. That was integrated genomics was all based in Chicago. But I was to come out to the West Coast, and near the end of '02, I came out and took up a role as general manager and SVP at a chemical diversity, which is a small molecule drug discovery library producer. So really a company of medicinal chemists. And ironically, that was supposed to be a 3-year project. And in 2023, I retired as CEO of that company. But in the meantime, one of the owners of Chemical Diversity and I decided to launch a private office, a private family office, to do investments, not my money, their money, in innovative biotech startups. So we did a variety of those, some in the pharmaceutical development space, some in health tech, digital health. And my co-founder, my founder, and me as the founding vice president, we developed a discipline of doing experiments in science and in commercial science to figure out how things worked and to try and come up with models to streamline and accelerate that work. So long story short, even though I'm retired, I'm still nominally the president of Torrey Pines Investment, and we deployed something north of $250 million over a 15-year period and had a lot of failures, had a lot of successes in a small way, but the net return on that was about 3x over that 15-year period. So that was encouraging. While we were doing that, and I was still developing chemical diversity from what was originally just a pure chemistry company into a chemistry and early stage pharmaceutical services company, including basically full stack preclinical and periclinical, and then ultimately operating clinical trials as well through phase 1, phase 2. So a pretty comprehensive portfolio of company services, and we were quite successful in growing that company about tenfold over that 21 or 20, depending on how you count, 21, 22-year period. And as I was preparing to leave at the end of 2022, we also began to stand up AI systems to design small molecule chemistry perform predictive analytics for docking studies and all the rest of that, where now ChemAI is a well understood, although rapidly expanding part of target validation, drug prototyping, etc., in an accelerated fashion. I can talk later about what we did in terms of that. But then from there, in 2018, I got curious about how venture capital worked. And so with that partner from Torrey Pines Investments and two others, we formed a general partnership called Teal Ventures. And I wrote the investment prospectus for that. Primarily, I thought that there were opportunities in medtech, and I still think there are opportunities in medtech, but I know a lot more than I did in 2018. But also because it was an area I was very familiar with, we thought that mobile digital healthcare technology would be extremely interesting as an investment proposition, because in 2017, 2018, that was only beginning to become possible. Little did we recognize when we founded Teal Ventures in November of 2018 and raised our first tranche of investor limited partner money that we were about to enter into a global pandemic, also known as COVID-19. So while in that first few months of at the end of 18 and the beginning of 19, we were actively investing in very early stage companies, seed stage companies in digital healthcare, they became extremely relevant when we had to shut down the world and everyone had to go virtual. We looked like geniuses. I absolutely disavow that. The major trends existed, but no one predicted we were going to face a global pandemic. But it became necessary to have those technologies. So that's going on. That was now it's 2026, and we have about a year left in that fund because it's a 10-year fund. And we're evaluating whether we raise fund 2 or not in Teal Ventures. As I already mentioned, at the end of 2023, I retired from working in chemical diversity, but still active in Teal Ventures and still active in Torrey Pines Investment. But in 2025, January, I still have an active mind and realized I was bored, and a new constellation of emerging technologies came forward, and I'll talk about it a little bit later. But what we're doing there is we're creating from the well understood mathematical and data handling, I call it data wrangling disciplines that enable omics, genomics, transcriptomics, proteomics, metabolomics, the big four. There are other omics, but those are the big ones. Those same disciplines can be used in the realm of physiology, and that meaning that electrical biology or bioelectricity. And so we're pursuing that in a company that I founded and co-founded called Flexion Therapeutics. What I want to build, what we are building, is the fifth, when I call it the missing omics, and that is physiomics. There's technology which allows you to interrogate cell membranes and ion channels and voltage and current activated surface receptors in mass, in omic scale data streams. We're building the quality control and data confirmation or standardization platforms that are needed to feed predictive analytics. And it's a different kind of mathematics in the AI from the other traditional omics, in that they're all built on linear maths, linear algebras, with rare exceptions, nonlinear mathematics. But physiology is intrinsically continuous variable math and transcendental functions. That's what's required. If you've ever taken courses in physics and electronics, you'll understand what I'm talking about. So it's a different kind of maths that we're using to build that AI. If time permits, we can talk a little more about physiomics and
Absolutely.
how I'm building that. But back to the questions. the series of doing science or business science as a business and exploring how things worked, being curious, we developed a discipline of doing small experiments. And I have some rules about how to do that. There aren't many, only about 3, but I'll share them a bit later. On how to understand an existing industry that has some bandwidth issues or some data quality issues or some time and money investment issues, which I think anyone who knows pharmacology and pharmaceutical development knows there are some three major issues that we face as an industry, and we'll get to those in a second. But
Absolutely.
to summarize those experiments that we've done both through Torrey Pines Investment and in Parallel Chemical Diversity and later Teal Ventures, there are 3 rules I have in this startup world. Number 1 is, in the startup world, do as many things right that you can. It is inevitable you will make mistakes. So that leads to axiom 2, which is, in the startup world, try not to do anything fatally wrong, because we can make decisions based upon incomplete information, which will kill our opportunity to create new technology. So it's important not to do anything fatally wrong. If it's a high stakes decision, then you need to consider it carefully and with as many experts as you can get access to. And number 3, always maintain a margin of safety. It's like gambling. Never bet the house on a single decision.
Absolutely.
So that's kind of in a longer way than I intended, generally explains how I arrived at the point where at the beginning of 2025, I was restless and still curious, and decided that it now was the time to stand up cell and membrane physiology as the next omics, physiomics.
Perfect. Yeah, I'd love to get into that a bit later. But looking over your career, you've definitely gone through major transitions from science to business, from private equity to more startups and venture capital investing to starting your own thing all over again in 2025. So, just to understand that better, who were you before you became the chairman at Fluxion Therapeutics and partner at Teal Ventures and Torrey Pines Investment? And how does that person still show up in your work today?
Yeah, that's a great question. I had a, as for an undergraduate career, I had a very good rounded education. At the time that I went to undergraduate school at the University of Iowa, the discipline, the major course of study of neuroscience didn't exist, right? At least at that university. It did exist on the West Coast. I could talk about that, but I understood that I was very, very interested in how the brain worked. But at the University of Iowa, there was no course of study that I could graduate in for. In my case, it took 5 years with a year out to work, but called neuroscience. So I kind of rolled my own degree. I took a degree in experimental psychology, a BS. Although originally I trained in clinical psychology, I realized I was much more interested in how brains worked rather than how psychopathologies worked. And then I knew I needed even more solid science background. So I took a minor in biology, a minor in biochemistry. And I knew that my quantitative skills needed to be better. So I took a minor in statistics, univariate and multivariate statistics. And then because there are a lot of questions in neuroscience about how the brain works, which are pretty much philosophically focused, I took a minor in philosophy. So I rolled my own degree. And as it turned out, I had a great break in the work study program because I had to work while I was all the way through school because I didn't have money, and for some situational reasons, I didn't have eligibility for much in the way of financial aid. But that didn't stop me. I was able to work and got landed. The first work study program I had was actually doing organ dissection in the pathology department. Not very sexy, but it built my dissection skills. But it
Yeah.
also paid the bills. So then I
Hmm.
landed the work study situation with Jim West in the Department of Anatomy, and I've already talked
Okay.
about what that
Right.
led to. So I thought I was pretty well prepared, and I was. I ended up, for personal reasons, at Northern Illinois University in Chicago, or outside of Chicago, where I connected with Jim Willett, Professor Jim Willett, who was an auditory neurophysiologist. And I thrived there. I was able to do electrophysiology in the auditory system, in the brainstem, and ultimately in cortex, and did some tonotopic mapping there. If anyone studied developmental neuroscience, you know that the way the sensory systems work is there's a representation of the physical sensed world that is three-dimensionally repeated in the brain structures. It's called visuotopic or somatosensotopic, or in auditory systems, auditory tonotopic organization of neurons, where they are, how they interact with each other, how they interact with downstream, meaning the further into the brainstem and into the cortex, and also back into the real world, the sensed world through the ears. The reason that and the reason that I started building Fluxion Therapeutics goes back to those days, because when I was doing single cell recording in the auditory brainstem, I knew that these neurons were interacting with each other in their local group. They were analyzing sensory signals, in this case auditory signals, in an ensemble fashion, both locally and then feed down backstream to the inner ear, the auditory neurons, and then upstream to the thalamus and to the cortex. So, but the technology for recording those kinds of if you break it down into an engineering perspective, multi-signal emitters, signal sources, and tried to look at how they interacted with each other. Multi-electrode arrays didn't exist. You had single, either glass pipettes for intracellular recording, or close adjacent to cell membranes, or metal electrodes, and you could measure spikes and all the rest of that. And so my dream was someday I would be able to take multi-electrode arrays and triangulate on the basis of near source position, deconvolve which signal emitters were and where they were located in a three-dimensional cluster. That math didn't exist. It wasn't just a matter that I didn't know it; it didn't exist. And it wasn't until engineers solved the problem of locating in the real world where a cell phone was located, based upon the relative strengths that it had as a signal in a cell phone tower or a cluster of towers. The math was developed allowing you to triangulate where in 3D space that particular cell phone existed. So that math is completely applicable to what we're talking about now in Fluxion Therapeutics. But in addition, the ability to handle these massive multi-source signals as an aggregate electrophysiological signal and deconvolving those, that math didn't exist either. But even more so, the ability to capture and what I call wrangle those aggregate signals didn't exist. It was through the discipline of building genomics, transcriptomics, metabolomics, proteomics, that the data management and informatics disciplines were built. And so and then on the back end, the storage capacity, and as we've seen, developing now multi-dimensional deconvolution of these very complex multidimensional signals that we get out of genomics, transcriptomics, etc. The whole discipline of what is called RNA velocities are built on the ability to deconvolve those signals, but it's discrete snapshots in time for all of the other omics. So it's as if you had a series of rapid single snapshots of a continuous process. Physiology is intrinsically a both time and event continuous process. So in order to get a clean, understandable flow of transitions from moment to moment to moment that you observe in dynamical systems, or in engineering, they call it dynamic response systems. Those disciplines now exist. And that was my practice, mine and my co-founders. That was our scientific and technological premise for building physiomics. Okay. So we're kind of there. You notice I keep coming back to physiomics because that's what that's my passion. But the origin of it goes all the way back to when I was a graduate student, and imagining that although I was using very primitive computers at that time, the imagination that someday we could actually interrogate that piece of biology in an n-dimensional way and really understand the dynamical processes that must underlie signals processing and ultimately cognition.
Yeah.
I'm happy to report that the neurosciences have provided us with far more complex and nuanced understanding of how brains work than they did back in the days when I was a simple auditory physiologist. Okay. So that's how I evolved. But then the second part of my personal development of how you can use capital, because at the end of the day, talent is a resource, time is a resource, capital is a resource. Time, talent, and money are fungible against each other. They are trade-offs between them. If you have all the money in the world, you can attack a problem intensively, but the limiting parameters are time and the available talent. And for me, the recognition of that relationship, that these three key resources are fungible against each other, led me to continue to inquire about how could we accelerate the development of new technology in my space, in biotechnology. How can we capture the best available talent for any part of the work that we're doing? And if we're developing along a value chain from an idea to a product, we need different skill sets. So that has implications for how you build your project teams, because project teams need to have different skills at different stages through their development. So we began to look at, especially at the advent of COVID, we began to look at how can you compose project teams such that they're flexible and you can use just in time and best available talent concepts for the next stage of development that you need to do. And then when those unique skill sets are no longer needed, because now you need, let's say, expertise in pilot scale pharmaceutical formulation and manufacturing, you no longer need the deep biophysical or biochemical or computational chemistry skills, right, to create the prototype drug. Now you need to produce quantities sufficiently large enough to do preclinical and then clinical studies, and ultimately to a finished dosage formulation and dosage form. Different skills required, completely different. The economics are very different. So we spent a lot of time using those to improve our ability to deliver them on time, on budget, at a cost that you could afford, because we were doing that work on a fee for a service basis as a contract research organization in chemical diversity. And as we moved forward, we launched my longtime partner and I launched an accelerator that allowed us to take some of these ideas, which if the listeners are familiar with large scale software development, these are exactly the same skills that Silicon Valley, the Bay Area, large scale software development teams throughout the world have now adapted. And that is lean, agile development, just in time project execution. So the pieces of a final product you need can be categorized into things which you must have, things which are good to have, things which are nice to have. They don't have to all be deliverable at the same point in time. As you move through that process, those goods to haves, the must haves have to be done. Then the good to haves will come next. And then the nice to haves will come as you get into later stages. The effect of that is you reduce the cost per unit of progress because you're not developing the things which you will need in the future, keeping in mind that as you're doing that development work, you have many risks you must meet. Risks like, is it technologically feasible? Will the marketplace be open for what you ultimately produce and will deliver in the pharmaceutical space as a new drug? There are many reasons why their failures can come along. There can be economic failures. There can be technical failures. There can be market obsolescence. So if you have a best in class, which is better than anything else, and you have the next best in class, you probably have closed the market to the third best, fourth best, and the Me Too drugs. Those are market risks. Now you're hearing me start to talk like an econometric person or an economist because I had to learn those ideas over time. But we experimented with taking time, cost, and talent requirements out of the value chain when those were no longer crucial. The organizing idea between all of those is something that comes out of NASA because when President Kennedy proposed that we go to the moon and deliver humans there and bring them back safely, almost none of that technology existed. NASA was a brand new U.S. government agency, but Kennedy set a task to the scientists and engineers of figure out how you can do this, right? And it was on the basis of that future thinking, they solved the problems and created the technologies which would be required in order to accomplish that big goal. One of the technologies they developed, and it's both a project discipline and it's a mathematical problem, is critical path analysis. So what we learned in our efforts in Torrey Pine's investment and then the I-2020 accelerator, which I use today, is this concept that when you think about the very starting point for a development of a new drug for a new disease indication, there's some things you must have. Obviously, if you've studied pharmacology and drug development, you know you have to have a validated target, and you need to have some chemistry or biology, which are tools for what a prototype drug would look like. But there are some non-scientific, non-technical issues that you must address. First of all, you have to have a well-defined drug product profile because that's what the the FDA consumes in defining what the regulatory pathway must be for approval. So you need to have a well-defined drug product profile. You need to have a well understood and mutually agreed between you and the regulator, the FDA in the US context, as to what must be accomplished and what data must be provided to the FDA in order to enable them to say, yes, go forward with your first in man trials, go forward with your clinical efficacy trials, phase 2, phase 3, your safety trials in phase 1, phase 2. And ultimately, when you file for a new drug application, what are the requirements for that data set? I call it a data dossier that you must deliver to the FDA, which gives them a persuasive case for approving your new drug. So that's the regulatory pathway hurdle. And then from there, back to wherever you begin, let's say it's at the point of you've fully validated this is the drug target which has a pivotal role in the creation or treatment of a disease, that piece of biology, to the final drug product profile. You can analyze what the critical path of steps you must take in your project plan. That critical path is the central part of what you must do in your project. In addition to that, we know that only one in, take your pick, 1 in 10, 1 in 100, 1 in 1,000 new drug products finally deliver a new chemical entity or a new biological therapeutic. How do you minimize those failures? Because it's a huge waste of the financial resource. You can improve the consumption, the efficiency of consumption of the financial resources by the disciplines you do inside the project, by focusing on critical path and these 3 buckets that I described: must-haves, good-to-haves, nice-to-haves, and sequence those so that you're not spending money before you de-risk the elements of that work that you would perform. And so that's where the critical path analysis comes in. And then finally, there has to be a development discipline. And the stakes are higher in biological therapeutics development or chemical therapeutics development than they are in software, because the cost per unit of effort goes up exponentially as you progress from early stage through preclinical, through periclinical, through clinical, and through final pivotal clinical trials, to the point that in total you're looking at an investment for per new drug that's delivered between $2.5 and $5 billion, right? The sooner you can de-risk or demonstrate that your path forward is not clear, the better off you are, because you're not investing money into futile efforts. So the discipline we developed for our lean, agile, just-in-time project execution requires a different mindset and a different set discipline, which erects go, no-go decisions early on and all the way through your project development. So there have to be high-stakes milestones. It's what I call progress or die. So if, in fact, you're blocked from going forward on a particular project with a particular therapeutic indication, with a particular prototype drug, you need to bite the bullet and say, we cannot progress further in this project. At that point, if you're unable to, fairly quickly, and they need to be repeated along each stage, you must have the discipline to either archive it or end the project. And this is a concept that overwhelmingly it's private money which finances these projects. It's private equity. It's investors who are sophisticated investors who understand the disciplines required to be successful. And they welcome this kind of a development plan where you have high stakes milestones, because they know that if you're not making progress, you're not simply treading water sideways and still consuming the financial resources, but rather you stop. And for whatever the reason is, you may archive it, you may simply sell it to someone else, or you may simply sink the cost that you have in it, which is far preferable to sinking $500 million into it before you do that, right?
Right.
So what does that mean? It means that the role of the project teams has to be significantly reconfigured. You have best available talent, and now that we operate in a distributed virtual world where your
Mhm.
best talent may be anywhere in the world and often is on multiple continents, right? Post COVID, that's not a difficult discipline to adopt, right? Where you're seated today and where I'm seated today, we're on opposite ends of the North American continent, right? But we're speaking basically with no time delays, no auditory delays, no technology delays. So we can effectively work together on a project. The second thing it does is it requires that the project team members' expectations need to adjust in that we had to adapt, and we actively do this in most teams now. I'd like to say we pioneered it, but it certainly was picked up by others and reinvented by others, where experts in a particular subdiscipline understand they will go along in this journey in that particular project they're involved with until their unique value add is no longer needed. And then they will move on to another project, and the original project will move on with others who have different skill sets. But the advantage is because we are a global development team, we can find the best available talent for the project we're executing at that point in time. The third key thing that has to happen is project managers who used to, in pharma companies, in CROs, full service, full stack CROs, and even specialty vendors, have to change their mindset so that they're not managing projects per se that are within their expertise, but rather project teams who, as a group, collectively have all the skills required for this phase of work. But then they also need to be looking forward to, in the next phase on the critical path, what skill sets will they need, and start working to build those teams even before they're needed, and then they onboard them as they're needed. That's where the lean, agile, just in time concepts that have been so successful in massive software development programs have been spectacularly successful and take out time, cost, and money, right? Time, cost, and available resources, right? Talent, money. And so
Absolutely.
that's the learning for why I'm doing what I'm doing, and how I got there was trying to solve those problems. I could talk about specific economic cycles and that sort of thing,
Mhm.
but it's not really necessary. I think the concept's the same.
Absolutely. Yeah, there's a lot of gems you dropped in there. I hope our listeners caught those. But Ron, I feel like you're one of those rare people in the industry who have experienced almost everything in it. You've been a scientist, you've been an operator, you've been an investor. So you've gotten a very well-rounded perspective on what the industry is like. So based off of that, what's the thing you're genuinely not happy about in biotech right now? And the thing that keeps you up at night, not because you're worried, but because you're just tired of it.
Well, I can talk about just tired of it because it was a pivotal shift in my career of leaving the lab as a research scientist and going into academic administration, which in the university setting is a very challenging role, underappreciated because you're immediately labeled as a bureaucrat, et cetera, et cetera, et cetera. But that leaving that aside, the reason I did that is the last published work I did, and that was at the University of Chicago after my 8th year, I think, in the lab, was a very long and grueling project, which, just to give you a flavor for it, doing correlated light and electron microscopy um, of this is in immunohistology that allowed us to definitively demonstrate the existence of two different transmitters in a single neuron, right? So this was a bilingual neuron. There's been a concept and a theory about this for many years. Now it's an established fact. We were one of the first who were able to demonstrate the existence of the synthetic enzyme for acetylcholine, choline acetyltransferase, and for epinephrine, noradrenaline, right, within a single neuron. And that required two immunohistochemical reactions in conditions which preserve the ultrastructure sufficiently so that you could, in a single identified cell, demonstrate the existence of both transmitters at the light microscopy level and then definitively at the ultrastructural electron microscopic level demonstrate that in fact they coexisted in the same organelles with subcellular organelles. It was a grueling work, very technically challenging, and it would take 23 hours from start to finish with no breaks. And once we finally had that, and it was published with the first author by Al Levy, who was one of the MD PhDs in the laboratory I was in at that time, who now actually, he was a great mentor for me too, but now he's the director of the Comprehensive Alzheimer's Research Center at Emory University. So he's been very successful staying in pure science. Okay. But and I want to give him the nod because he wrote the paper. Be that as it may, I was done with bench science. I had just reached the point where I fell out of love with it, right? And I never thought I would fall out of love of science. And it turns out I didn't. That particular piece of work I was done with, and I wanted to live a different way. So that was one. The other question you asked, did you ever nearly walk away? So that kind of answers that. But I didn't, because I stayed deeply involved in the NIH and other agency-funded graduate training programs there at the University of Chicago, the MD PhD program, developmental biology, cell and molecular biology, immunology, a number of large programs where when I was in the Department of Molecular Genetics and Cell Biology, I was involved in co-writing those research grants and then the training grants as well. So I stayed, but I stayed in an indirect role as a science facilitator role, let's call it that. And then from there, you can fill in the rest of the blank of why after we built the resource center at the University for whole genome sequencing, decided to go try and do that for fun and profit. That keeps me up at night. I think, and I have written a blog piece about this that you and I discussed prior to this session. I'm pondering whether to publish it or not. I think that we have a tremendous lack of discipline as drug developers in the industry as a whole, and in being realistic and really brutally honest about the prospects of projects that we're attempting to create new drugs which are intended to be an improvement on a very effective standard of care. That's this problem of Me Too drugs, of Me Better drugs. And the reason why I get annoyed about it is because by conservative estimates, and some of these come out of Tufts University, the Center for Pharmacoeconomics and other research centers, there are between 1,500 and at the high end, maybe 7,000 when you include rare and semi-rare drug or disease indications for which there is no standard of care. There is no widely or universally recognized effective pharmaceutical therapy. That means there is a huge unmet need. The problem is one time. The way we develop drugs, it takes far too long, between optimistically 10 and 17 years from inception and launch of a development program to, except in the case of some oncology drugs, and even those are getting longer and longer because the oncology drugs that are being developed today are much, much more sophisticated than they were when I stepped into science. It embarrasses me to say it, 40 years ago. And in addition, so the discipline's not there for lean, agile, just-in-time, critical path, flexible project teams, all of these disciplines or discipline aspects that I think we should be using. And the other is that we are not focused on eliminating time, because much of the time in our drug development process is effectively wait time. It's as if you had a AI capable laptop which had a mandatory 30-second wait interval between one AI step and another. And it's artificially introduced because of the discipline that's used in drug development. And it seems on its surface legitimate. It's treating each CRO that you need to hire as a unique vetting process. There are many, many highly competent CROs out there who understand their particular niche and their unique value add. For financial reasons, they aspire to get more of the value chain and perform it themselves. But with the models that we've developed, especially through I-2020, and now I'm going to mention a different area, a different initiative that I'm not directly involved in, but I was involved in the early part. That's the LOCH projects, LOCH projects, which were an outgrowth of the work we did at the I-2020 Accelerator, except the innovation there is to have a private equity investor partner who are completely dialed into this lean, agile, just-in-time project discipline and has sufficient capital to take projects through when they meet the filter for onboarding projects, to finance them in the millions to multi-million-dollar level to take them to the next major value inflection step. So what does that allow you to do? It allows you to address the capital need, that limited resource. It's not unlimited, but it's enough so that you can move through these high stakes milestone inflection points. And by doing that with a stable, a portfolio of external development partners, CROs, you have them already lined up and pre-vetted so that you're not spending 6 months to a year looking for the next best CRO. And that takes that critical time of no development out, largely eliminates it from your development perspective. In that project, and I don't claim ownership of it, my partners, Nikolai Sovchuk, who's just to drop a name, Orbimed Advisors as the financial partner, they have created more than 30 single asset companies. And on average, from inception of the project to first in man studies, they've been averaging 3 to 5 years to first in man study. And generally, depending on the therapeutic indication, from a $5 million commitment up. And that is an order of magnitude lower than what most others are doing. I know that Roche has launched a similar, I know I'm dropping names, I probably shouldn't, but so let me anonymize it. Several integrated global pharmas have begun to adopt that same kind of approach.
Mhm.
And the reason it upsets me that we don't do this universally is that I know that there is a way to do this work which would allow us to take the delivered sunk cost of a new drug from $2.5 billion to maybe $1 billion, maybe less than $1 billion. And what that allows us to do then is, since the financial resources is a limiting parameter, it allows those dollars or whatever currency, that investment to be spread across a larger number of high value, urgently needed new therapeutics. Because if you do some simple arithmetic and you need, let's say, minimally 1,500 new drugs to address the lowest estimated number of unmet need therapeutically, that's 1,500 times $2.5 billion. That's approaching the U.S. domestic gross product, right? Right. And so then if you replicate that across all the countries that need new drugs, and maybe we say it's 7,000, it's about equal to the GDP of the entire planet.
Hmm.
We can't do that realistically. So how do we do it? And from my perspective, it's only through these kinds of lean agile disciplines that we do that. So that's why that keeps me up at night.
Mm.
But
Right. Yeah.
but I'm not the key decision maker in that anymore. So I've made a conscious decision, and I may be involved in it at some level in the future, but I made a conscious decision to try and create physiomics and demonstrate that physiomics as one of the five fundamental omics deserves a place at the table in national policy.
Absolutely.
And for Fluxion Therapeutics, we've also made a decision, myself and my co-founders, that we are building the first full R&D platform for that in Ireland, because Ireland has set a national priority to become a leader in AI-driven, AI-focused, large-scale pharmaceutical development and biotechnology. And they've aligned their national financial and economic policies to support that.
Wow.
So we have a company named
Hmm.
Fluxire, right? And we're in the process of standing up the website for it, but it's the same, excuse me, it's the same concept. And we're using these principles I've described for building that company. So stay tuned. A couple of years from now, I hope to have
Absolutely.
more to say about that.
That's great. Yeah, we'll have you back on when things are on track with Flexire. I love the name. It's absolutely great, and it's it really comes back. It's like a full circle moment to see where you started, how that's involved in Fluxion, and now how you feel like it's a problem that keeps you up at night as well. So it's great to see that alignment within your answers. And just looking at that, you seem like a very aligned person. You know what you're doing. You know why you're doing it. So just based on that, let's say an article got published today. Ron's 5 rules to having a successful career and satisfying life. What would those 5 rules be?
So I gave you the three short principles, right?
Yeah, the
But
three axioms.
that fits the startup world. As you know, we talked about this previously, because I was able to present some of these ideas to the Nexus Club at USC when you were coming to the end of your undergraduate career, right? And we've talked a little bit about what's next for you. I think that's very exciting. But yeah, so I have 10 rules. If you accept 10 rules,
Yeah,
I can.
10's
All right.
perfect.
All
The
right.
more the better.
Right. So if I were planning my career, I I did actually learn these along the way, but I didn't know them coming into my plan to become a pure research scientist, because the practical applications kept coming up to me. So I had to follow those, and that's it. Number one is stay curious. And I think that's critical if you want to evolve in your career. And I have down at the bottom, I have the reason why it's important to be flexible, which is my number two. Because the career you've trained for, at least for me, the career I trained for has evolved beyond my skill set. So I've had to continue to build my skill sets as the opportunities arose, as this focus on creating drugs more rapidly, or creating, once I got out of the academic space, creating new useful tools more rapidly and at a lower cost. These are the things I had to learn. Stay curious, stay flexible. Number three, and this I owe to my mentors, all of them in academia and in business. Learn what critical questions are and how to ask them. And if you don't know, ask somebody who knows,
Hmm.
who's demonstrated it. And this goes to my number four, which is, in every situation, find and cultivate mentors. Don't try to invent everything again. Use what people have already learned for your own need. And that especially matters for things where you find that you want or need to know them. Find mentors. Don't try to study it all on your own. Number 5 is they used to call it the Protestant ethic. Dig in and do the work.
Mm.
All right. Just go in there and get your hands dirty, roll up your sleeves, do the work. Don't wait for others to show up and do it for you. If someone ever has asked you, why did you never do X? Don't let your answer be because nobody showed up and did it for me. Right. And that's why you need to continue to learn things. Recognize and focus. You need to understand yourself. Understand and focus on what your unique values are. What is your unique value add? All right. And then instead of waiting for people to show up to do the work, find the people who do what you're not good at. And or they have unique value add, and be humble enough to recognize that. Number 9, practice evolving business models to solve industry problems. And those 1, 2, 3, 4, 5, 6, 9 words, I think, summarizes what my career has been, successfully or unsuccessfully. Because I've had many, many failures. I've tried many things that didn't work. Right. But that comes with the work, the territory.
Absolutely.
And then the bad news. The bad news is the advent of AIs and agentic AI are commoditizing our specialized technical and scientific professions. And this is the next imperative as to why we need to continue to be lifelong learners.
Right.
So that summarizes what I can point to that's made me successful.
Yeah. Ron's 10 commandments. Absolutely great. Yeah, those are very comprehensive, very easy to follow. And I feel like there's a lot of reflection that must have happened for those to boil up.
Yeah.
And so one more very reflective question, and the last question that I have for you is, when you look back on your career years from now, what do you hope your legacy will be?
Okay. Well, obviously today, I hope my legacy is that physiomics is recognized as being the powerful connector across all of the omics, the systems level. And this is ultimately what I want physiomics to evolve into, is a fully descriptive view of systems biology, out of which network pharmacology, all the other network omics naturally arise. Because if we go all the way back to when we weren't here when it happened, when the first single cell organism happened, however it happened, the way it became possible was the semipermeable membrane. Because semipermeable membranes allow you to segregate charge between the inside and the outside of that single cell organism. And today, every cell that we know of, if it has a metabolism and even very viral packages, right, do segregate charge. And it is that electromotive force which drives the chemomotive force, which drives everything from energy production in the oxidative phosphorylation, electron transfer chain of mitochondria, all the way up through membrane or voltage gated GPCRs, which after ion channels, by the way, I haven't talked about ion channels, but after ion channels is the single richest source of new therapeutics we've had. But keep in mind, 9 out of 10 new drugs over the last, choose your 100 years, whatever interval you want, exert their action through ion channels. So when I talk about physiomics and I talk about Fluxion therapeutics, I'm really talking about ion channels, which are voltage and current gated organelles, and the context in which they operate, which is the bilayer lipid membrane. And as a group, going back to my dream of being able to understand how auditory physiology, auditory signals get processed, we now know that the cellular membrane is not a uniform lipid bilayer, but rather is a highly differentiated and dynamically changing biophysical entity whose function and mode of making a living is really segregating and moving charge inside and outside of the organism. And the same thing's true with subcellular organisms. So the return of biophysics.
Absolutely, a full circle moment.
So that's one. The second is, yeah, I would like to be able to say, with respect to the drug development paradigm, I had some small influence on how it was changed to respond to what the human need is. And the other one is a soft legacy, and that's that people can say, well, this is someone who pursued his vision with integrity. That's it.
Absolutely.
I think in terms of scientific leadership, it's absolutely critical that we be mindful that as CEOs, as chairman, as officer of a corporation, or a head of any nonprofit organization, large or small, we owe a debt of integrity to the stakeholders, and that's been how I've guided my own life. Enough of that. So
Yeah. Yeah, no, that's a very beautiful legacy, and I hope that we're able to see it, and we are slowly seeing it already. Thank you so much for your time again. I really appreciate you taking some of your time and leaving behind this legacy with It's Bigger Than Biotech. I really appreciate that. And yeah, again, thank you so much for your time, and for our listeners, thank you so much for listening to It's Bigger Than Biotech. See you on the next episode.
I love the title of your your podcast. It's obviously you and I are highly aligned, and I'm really grateful for the opportunity to make these remarks. Thank you very much.
Thank you.